So I'd like to introduce Anne Churchland, who is going to tell us about spontaneous movements dominate cortical activity during sensory-guided decision-making.
Okay, thank you very much for the introduction, and thank you all for being here today. It's really been a great privilege to be one of the co-organizers of this meeting. We had a really fun time putting it together, and it's amazing to see what the turnout has been. One of the things that's been especially fun for me is that there's a lot of really young people in the audience. Can you raise your hand if you're a student or a postdoc? So huge turnout from people who are trainees, which I think is great, and I really want to echo Matteo's sentiment to encourage you to please ask questions at the end of the talk. I know this can be intimidating because if you ask a bad question, that can happen. It's a possibility, and you feel bad, right? And I know this because once at Cosine, when I was a postdoc, I did ask a bad question. I was so nervous, I kind of couldn't get the words out, and I asked something that didn't really make any sense. And does anyone here remember that bad question that I asked at Cosine? So, okay, this is good news. Even if you ask a bad question, only David Heeger will remember, and nobody else will remember. So go ahead and ask questions. So I'm going to tell you today about some new work that's just coming out of my lab based on some new techniques that we've been developing in the lab. And because this is a really diverse audience, I'll start off by telling you a little bit about why we study decision-making in mice. And we do this for a number of reasons. One of them is we consider them to be a super cool and interesting model organism for a few reasons. So first of all, mice are predators, and they use their visual system to localize moving prey. This here is a mouse, and this is a cricket. And this was an experiment designed by a postdoc, Jennifer Hoy, in Chris Neal's lab. And she demonstrated that mice will hunt crickets. So it's chasing it around an arena, and she's demonstrated that this is truly a visually guided behavior by blocking other sensory modalities like audition and vision and so on. So mice, in addition to being predators, are also prey. So that means they also use their visual system in quite a different way. And that's exemplified by a stimulus that was developed in Marcus Meister's lab. It's called a looming stimulus. It's a circle that gets bigger over the mouse's head. I guess it's reminiscent of a hawk. It looks like this. And it causes the mice to elect to either freeze or to run for cover in a little shelter off to the side. So a postdoc in my lab, Ashley Jravenet, has developed a multisensory version of the stimulus. I'll play that for you now. So it has a sound that's growing in amplitude in parallel with the visual stimulus. And this is what happens to mice when they experience that stimulus. I'll show you that one more time. So this demonstrates that mice are not just using their visual system to track predators, but also to avoid prey, quite different functions there. Also, an interesting feature about mice that makes them particularly fascinating for study of the visual system is that they are not nocturnal. So they lack a tapetum, which means they're not a truly nocturnal animal, and they either are awake at night or awake in the day, depending on the availability of food. And to me, this is an interesting fact because it points to a visual system that has to be used flexibly in combination with other modalities. So if you're a mouse out in the daytime, the light levels are high, your spatial resolution is not too bad. Whereas if you're at night, kind of going around somebody's apartment looking for cheese, the visual systems are not as good and you have to rely on other modalities more. And so this ability to flexibly use visual information alongside other sensory modalities points to a Bayesian combination of incoming sensory signals that may even be optimal. And indeed, in rat studies in my lab, we've showed that animals indeed use those signals in a very judicious way. Another super important reason that we study mice in the lab, and will really be the focus of the talk today, is that they're transgenic animals, so there's lots of really interesting genetic tools that we can use to measure and manipulate neural activity. This is an example image from a paper written by my colleague at Cold Spring Harbor, Josh Wong, where the inhibitory neurons can express a fluorophore so that you can identify them in the brain. And a postdoc in my lab whose work I won't have time to talk about today, Farzaneh Najafi, has used these mice to understand the role of inhibitory neurons in decision making. Other kinds of transgenic animals are available that are relevant today are the ones that you've heard about in some of the other talks throughout the conference. In particular, mice that have calcium indicators that tell us about neural activity, give us a measure of neural activity. So the mice that I'll talk about today, this is work done by Simon Newsall and Matt Kaufman, who both were postdocs in my lab. Matt left a few weeks ago to become a professor at University of Chicago. And they used mice expressing GCAMP and all the excitatory neurons. And so the upshot of that is that when the neuron fires, the neuron gets brighter, and we can capture that fluorescence in different kinds of microscopes. And I'll tell you actually about a few different setups that we're doing today. So most of the important work of this talk will be when we use those setups to give us an indication of ongoing neural activity. We also use that fluorescence signal to measure maps of visual space within the visual cortex, and you've seen these previously throughout today. This is primary visual cortex, and this is about 5 or 6, depending on how you count, surrounding visual areas, and each of these has a unique map of visual space. So the mouse, just like the human, is carrying around multiple maps of visual space within its visual cortex. And this is both an interesting phenomenon in and of itself and also a super useful tool. We can make these measurements through the skull and very easily, because the mice are lissencephalic, they have a flat cortex, so it's quite easy for us to visualize this compared to in other animals where the cortex has a lot more bumps and grooves. So in the behavioral paradigm that I'll tell you about today, we have animals that are doing a two-sided detection task, so they're presented with either an auditory or a visual stimulus. It can be either on the left or on the right, and they have to lick to the side where the stimulus was present. So if there's a click on the left, they lick to the left. If there's a click on the right, they lick to the right. So one key feature of the experimental design that you'll want to keep in mind, in addition to the fact that it's visual or auditory, is that we decided to train separate cohorts of mice to be experts in each of these modalities. So in cohort 1, they're experts in auditory decision making, but they're pretty much at chance on the visual. If we gave it to them longer, they'd figure it out eventually, but it takes some time, so we take advantage of that early training moment where they're great at audition and at chance at vision. And then we have a separate cohort that are the opposite. They're experts in the other modality. And I'll show you their performance in a moment. But this turned out to be a comparison that was rather surprising, and I'll tell you more about that in a moment. So here's the structure of the task. There's a baseline period, then the animal is going to grab a handle to demonstrate to us, indicate that it wishes to initiate the decision. There's then a stimulus period on which the visual or auditory stimulus appears, disappears, then appears again. Presumably at this point the animal knows what it wants to do, but then it has to wait a little bit during a delay, and then finally it's allowed to make a response. So I'm going to show you some videos of some of those moments in the trial now. So this is an image of a mouse. It'll be a movie in a second, and you're seeing a camera position underneath the mouse. And that's because it's reporting its choices by licking to one side or another, one lick spout or another. So we need to see the mouse from the underside so we can see what side it's licking to. And these down here, these are the two handles that the animal will grab to initiate the trial. And these are the lick spouts that at the moment, they're controlled by servo motors, but they're positioned away from the mouse so that it can't report any decisions yet. Okay, so the servo motors bring in the handles, and should the mouse wish to initiate a trial, then he will grab the handles. Okay, here's the stimulus period. You can see on this particular trial, this is now, I'll show you one more time, the camera is now from the mouse's back, so we have two cameras, which is going to turn out to be important, and you can see the visual stimulus on the right, and the animal has to wait before making a decision. You'll notice he made a couple of spontaneous, funny movements there that we didn't instruct. Keep those in mind, those will be important later. And then eventually, this is now the other view, the animal makes his response. And the servo motor that he didn't lick to moves corresponding to the spout that he didn't lick, moves it out of the way. Okay, so here's the behavioral data for the two cohorts of trials. And overall, mice can be trained to be pretty good at this task. We like to have a pretty quick pipeline so that we can first map their visual areas and then image them during decision making. But many of them get to be about 90% correct. The average is about 80% correct on their expert modality. So these guys are expert at vision, pretty much a chance on audition. And then this is the cohort, or the opposite. They're experts at auditory, and they're a chance at vision. So neither of the animals are perfect even at their expert modality, but what I hope we will agree with is that there's a big difference between the two cohorts. The behavioral difference between these guys is quite evident. Okay, so armed then with mice in each cohort, we had 6 mice in each cohort. We imaged them under a wide field macroscope, which is the same scope that was used to generate the maps of the visual cortex that I showed you a moment ago. And this is 2-channel, and the reason it's 2-channel is that this allows us to get rid of the hemodynamic artifact that can make wide field signals hard to interpret. So Simon built this macroscope and is able to image the mouse brain through a cleared but intact skull. So we don't have to make a window, and we can just image directly through the skull because mouse skulls are fairly thin. And we do this in EMX-Cree CAMK2 alpha mice crossed with AI93 so that they have GCAMP in excitatory neurons. Okay, so now I'm going to show you some imaging data. This is just an image of the brain by itself through the cleared skull. You can see we can visualize it pretty well. And then I'm going to overlay the Allen Institute areas on top of it. This is the baseline period. Animal grabs the handle. Stimulus comes on, V1 is going crazy. This is a visual stimulus. The second stimulus comes on. Then there's a delay. V1 cools down. And then a big response. So I'll play that one more time. Okay, so this movie is slowed down considerably, but I hope what's evident from the movie is that we get lots of cortical modulation throughout the task, and that some of it is in the areas that you would expect. And I'll show you a few more plots to kind of give you reassurance that we're seeing activity in the places that we'd expect to see activity at the moments that we would expect to see it. So here's sort of a summary of the movie that you saw a moment ago, again overlaid with the Allen Institute areas on top of it. These are the different periods in the trial. And the first comparison that we made was just comparing the performance on auditory trials versus visual trials, just pooled across all animals. Super straightforward. We see a much bigger activation in area V1 on visual trials compared to auditory trials. Exactly what you'd expect to see. Pretty much the same with retrosplenial, which is sort of thought to be a secondary visual area. Not too much is known about it, but again, sort of reasonable. And then in these more anterior areas, the hindlimb, somatosensory, and also secondary motor cortex, we tend to see not much of a difference between auditory and visual trials. Again, this should provide us reassurance that most of the variability is coming from where we'd expect it to, because we don't expect the responses to be very different on auditory and visual versions of the task. Okay. But then this, to us, was a big surprise. So the reason we came up with this experimental design of having the novices and the experts is we thought, well, what do we want to do? We're a decision-making lab. We want to find decision-making areas. And here we have these two cohorts of animals. And let's say we're showing them all a visual stimulus. One group is really making a decision. They're right almost all of the time. The other group is not making a decision. They're making a sensory motor guess. So all we have to do is compare the neural activity between the novices and the experts, and whatever areas are different between those two cohorts of animals, clearly that will be a decision-making area, because these two groups differ only in the fact that one is making a decision and one is not. But this turned out to be a lot harder than we thought. And when we compared trained versus untrained, novice versus expert, we found that in sensory areas, secondary areas, and these more anterior areas, that things look pretty much the same. So this was a bit of a frustration for us as a decision-making lab, but we were not daunted, and we thought, okay, we need to take a closer look. We need to clean up the signal. We've learned from the work of other people that I'll mention in a moment that a lot of neural activity can be kind of corrupted by motor signals, other kinds of signals. Maybe if we build an encoding model to figure out just how much impact those are having, we can kind of get rid of those, clean up the signal, and finally see what we would consider to be decision-making signals, and then we're going to study those. So this is what we did. We did the natural thing, which is to build a linear encoding model. And our goal was to model the trial-by-trial fluorescence, which is over here. And so we're trying to find a set of weights that is going to map all these different stimulus regressors, which we put in a design matrix, to the fluorescence signal. Okay? So the design matrix is super large, and it includes a lot of the traditional experimenter-imposed variables that you might think of, right? Post-event things like the moment that the stimulus came on. Peri-event would be things like the moment around the time the animal licked for a choice. These are whole trial variables, like for example, whether the current trial or the past trial was a success or a failure. And then we included these analog variables, which are actually quite numerous, but are shown in a squished version here. And those included two kinds of things. So the first kind was traditional data that we got from the video, things like the size of the pupil. People are starting to realize pupil diameter matters. We thought, okay, we'll throw that in. Other kind of video regressors like whisking and licking that we had some kind of prior idea might be important. And then we took all of the remaining video data and orthogonalized it to all of the other regressors and kind of included that as an agnostic regressor. Sort of everything that was in the video that might matter for the fluorescence, but that we didn't happen to have thought of a priori to include in the model. So this is in red is the actual output of the model. And I should say we fitted this with ridge regression, so we were trying hard not to overfit. And you can see that we don't fit these little itty-bitty calcium transients here, but we do fit the overall signal quite well. We capture about 30% of the variance. Okay, so this is just showing you a little bit more about the analog regressors. This is the pupil signal, which we measured with one of our cameras. You can see that the pupil size goes up and down a bit. We have to do some work to clean up the signal, which I won't have time to tell you about today. Whisking was another one that we included. And these regressors were inspired by Marius Pachitaryu's talk last year at the Cosine conference. We also had a piezo sensor for body motion and also included handle, touch, and licking, and that was motivated by Carsten Strigger's poster last year, 2017, at the Cosine conference. So overall, the model worked pretty well. We were able to capture a lot of the variance in the fluorescence signal using this model, about 30% of it overall, and this was true throughout all of these different trial epochs, and we did this on cross-validated trials. So then we went and said, okay, well, which are the regressors that are really doing the work? What can we learn about decision-making signals by looking at which regressors really matter to the model? And there's 3 types of regressors that I'll tell you about, and the first 2 are in the category that I would consider experimenter-imposed regressors. So these are cognitive things like the previous choice, current choice, expected reward, and so on. And you can see these captured a certain amount of variance in the model. Sensory things like the onset of the visual stimulus, onset of the auditory stimulus. Again, these captured some variance. And then the final category are motor variables. And so these include all of these different movements that you see here that we got directly from the video. And then this one labeled video is all the other stuff that was left in the video that was not, that's not captured by these individual motor regressors. So what I hope is evident is that this category here, motor, captured way more of the variance than any of the other regressors. So I'm going to refer to this category of regressors here under motor as the internal backdrop, and that's for a few reasons. First of all, it's to kind of distinguish it from these experimenter-imposed variables that we pick and that we think are important. These are either learned or spontaneous movements that the animal has elected to engage in. And importantly, even though what we're measuring are movements, these might also be indicators of internal state transitions in the part of the animal. So for example, even though the pupil captured a fair bit of the variance, not actually as much as we thought it was going to, but it was certainly an important regressor. It may not be that actually there's that much cortex devoted to controlling the pupil, but instead the pupil becoming more or less dilated reflects an internal state change, becoming a more or less aroused, for example. Okay, so this was a surprise to us that this internal backdrop mattered so much, and we were somewhat skeptical of this, and so we wanted to have kind of a more conservative measure of the importance of these different regressors. So what we did was to assess the single regressor contribution. So this is a schematic of the full model here that I just showed you, and then we take some regressor that we want to evaluate, and we just dump it out of the model, and then we ask how much worse did the model get. And this is really the most conservative test of a particular regressor because it might turn out, it might look like it has a lot of weight, but then when you kick it out of the model, the other regressors end up taking up, capturing up a lot of the variance, sort of sopping it up anyway, which would tell you that it's not actually important. So this is a more conservative thing to do. When we did this, this is telling you kind of how the model suffered when we threw out different parameters to reassure you that this is a reasonable thing to do. Okay, so this is when we threw out the right visual stimulus. This color bar is the delta R squared, so kind of how much the model suffered, how much worse the fit was. And you can see when we throw out the right visual stimulus regressor, the model gets worse at predicting V1. Okay, makes sense. Here, if we throw out the right handle, the model gets worse at predicting the forelimb somatosensory area and also the mirror image of the forelimb in primary motor cortex. So the unexplained variance is fairly well localized. We then did this for all of the regressors, and this is the same plot that I showed you before, except now in white on the inside. Now you can see how much there was how much change there was in R squared, how much worse the model did if we threw out that particular regressor. So let me just contrast 2 different regressors here. Over here, this is time. So when we include time in the model, this is just like a 1 for every moment in time, it seems like time is really important, like maybe the animal has a sense of the passage of time or something. But then when we kick that regressor out of the model, it doesn't really change the model fit very much, presumably because this time can be sopped up by a number of the other experimenter-imposed variables that happen to always occur at stereotype moments in time. Okay, but by contrast with these motor regressors, they tended to change less, and overall, this video, the orthogonalized video data, really was quite well preserved. And this regressor here, importantly, this is not a task-aligned variable. So all of these internal context variables, these are all aligned to the task. This one is not. It's orthogonalized to all these other regressors. So what this means is that there are a lot of spontaneous movements that we didn't tell the animal to do that have nothing to do with the moments in the task that are having a big impact on the neural signal. And this was a big surprise. Okay, so this is a summary of what I just showed you before, except I've grouped these things. I've just sort of collapsed across each of the categories. So this is sensory, cognitive, and motor. And you can see what you saw before, that with motor, the orthogonalized video data accounts for a lot. So animal movement accounts for about 27% of the variance in the data, and about 12% of that variance is the task independent. These are movements we didn't instruct that the animal elected to do. Okay, and this is just showing you where that unique variance mostly is. And it's a lot of that variance is mostly in the frontal structures, as you might imagine, because that's where a lot of the motor structures are located. But importantly, a lot of the unexplained variance is in sensory areas as well, which was a surprise. Okay, so what does this mean for the PSTH? So I've told you a moment ago that a lot of a substantial chunk of neural activity is really described by spontaneous movements that the animal selected to do that we didn't know anything about. But what's the consequence for this as an experimenter who really wants to understand not just spontaneous fidgets, but also movements that are related to a task and possibly some of these sensory and cognitive signals as well? So to do that, we take a look at some of the trial average data, for example, from V1, and then we can look at the impact of individual groups of regressors, for example, task, which is sensory and cognitive, versus motor, and just see how much of the true fluorescent signal in gray is accounted for by those different groups of regressors. Okay, so here in V1, you can see that at the moment that the stimulus is on, that's these gray bars here, that the task regressors in the model account for almost all the variance. That's good, it's V1, it's just what we would expect. Although notice, there's this gray bump here at the just preceding the visual stimulus. There's nothing on the screen at that particular moment, but there is a big bump in fluorescence activity that the task variables don't account for at all. But over here, this is the prediction of the motor variables, and they account very well for that gray bump early on, presumably because it's a movement that the animal's making, and it's not reflecting a sensory signal, even though there's a response in V1. Okay, so now I'm going to show you the same thing, except this time for area HL, which is a hind limb area. And this is really interesting and made me quite fearful about interpreting decision-making signals. So again, the full model accounts for the trial average data pretty well. Here are the two visual stimuli, and the fluorescence in this area sort of ramps up during the time that the animal is forming its decision about the visual stimulus. Then it's kind of flat during the delay, and then there's a big burst around the time of the movement. But interestingly, none of those temporal dynamics are explained by the task variables. So there might be any of a number really of interesting explanations for this ramp up. Maybe the animal's accumulating evidence, maybe it's an urgency signal, maybe it's could be any kind of interesting cognitive signal that has a time-dependent rise. There are a lot of them. But in fact, that time-dependent rise is really explained by upcoming and also current movements that the animal is making. So I think this part is kind of really a cautionary tale and should remind us that when we see signals that have interesting temporal dynamics that are reminiscent of what we might expect from different cognitive processes, to think deeply about how ongoing spontaneous and instructed movements might be affecting the signal. Okay, so the next thing we did is we said, well, we haven't given up on this idea of finding a decision-making area. We'd like to stay a decision-making lab. So let's kind of look across the brain, and maybe we can find some areas where there is at least some modulation by the task, okay, where these task variables seem to matter. And then let's actually zoom in a little bit. So this is what we call a task index. It has to do with combining an area that's modulated a lot and also with an estimate of how much that area is modulated by the task specifically in this conservative way that I described. And you can see that there are a few areas, so V1, some areas up here, and also area ALM, which is an area that's been implicated both as a motor preparation area, but more recently as having some interesting temporal dynamics that have to do with sensory processing, decision-making, motor preparation, and short-term memory. The neurons there can be active for a while as an animal is holding a plan in mind during a delay period, for example. So what we did, and this will be, I think, reassuring for anybody who's skeptical about the wide field signal, as in fact, perhaps you should be, because it's reflecting neural signals that are pooled over many, many neurons. We decided, well, why don't we dig a little deeper into area ALM and to redo this entire approach, but this time using 2-photon data instead of wide field data. So we took the same animals that we had just looked at in the wide field setup, and we moved them over to our 2-photon microscope, and we imaged them there. And we identified the neurons for this particular study using Sweet2P, which was developed by Marius Pachitariu. This is where area ALM is. It's actually the right ALM. You can see we can get lots of individual neurons in our field of view at the same time. And this gives potentially a really different picture, right? You might have wondered maybe the predominance of motor activity that I reported to you a moment ago had to do with the fact that we're measuring wide field signals. Maybe wide field is a weird measure, or maybe these motor signals are just teeny tiny in single neurons but are shared across large populations, in which case they shouldn't really be very evident or should be much weaker at the single neuron level. So fortunately, with the 2-photon, we were able to put this to test. So here are the individual neurons. Here are just an example fluorescence traces, and here are some PSTHs of individual neurons. If you're an ALM aficionado, you'll be reassured to know that a lot of these neurons are direction selective around the time that the animal makes the choice. This one prefers left licks over right licks. Same with this one. This one's active a little bit earlier in the trial, quite similar to what other people have reported in ALM. Okay. So many neurons show clear task tuning. So now we were in a great position to put our hypothesis to the test and see what happens when we look at individual neurons, and we found exactly the same thing. So once again, we found that cognitive and sensory variables, these experimentally imposed variables, were important in explaining the fluorescence signal, but not as important as movement signals, and certainly not as important as this motor regressor over here, the video regressor, which reflects non-task aligned, spontaneous, self-initiated movements by the animal. And this is just a summary plot here. I think I'm going to skip this slide. I'm almost out of time. This is we ordered the neurons based on excuse me, their R² in the full model, and then we looked at the individual contributions. You can see there's a lot of variability across neurons, and that sometimes you might have a neuron that seems like it's totally unmodulated, like its PSTH is essentially flat. Like this neuron here, it kind of seems like this is an untuned neuron. Normally people would just kind of throw this out of their population. Oh, well, this neuron, whatever, doesn't do anything. But actually that neuron does do something. It's explained very well by the full model, and that's because it has a lot of this non-task aligned motor signal. So that neuron, it shows up on your PSTH as being flat, doing nothing, but really it's responding to these animal selected self-initiated movements. Okay, so in summary, cortex-wide neural activity during auditory, we studied cortex-wide neural activity in auditory and visual decisions in mice, and cortical responses were surprisingly similar between novice and expert mouse decision makers. Self-generated variables vastly outperformed task variables in predicting neural activity. And finally, a linear model revealed previously hidden task-related activity in single neurons. So these are the people that did the work. This is Simon Musil and Matt Kaufman, who are listed here. They were assisted by 2 technicians, primarily Stephen Gluff and also Hien Yuan in the lab. We're also really grateful to our funders, especially the Simons Foundation. This work was critically supported by the Simons Collaboration on the Global Brain. They supported Matt with the postdoctoral fellowship that made it possible for him to do this work, and also made it possible for us to get the equipment to build the wide field setup and also parts of the 2-photon. These are really new techniques in my lab. We typically did electrophysiology, so imaging was new, and we really required support to be able to do this innovative work that would not have been possible otherwise. So thank you all very much, and I'd be happy to take questions.
So I see one right here. Okay. I think you already asked the question. Did you already ask a question earlier today? Yes. Do you mind passing the microphone ahead then? You ask a question too. Give it back then. Okay. All right.
Have you tried trials without a delay period, and do some of these spontaneous movements occur during the delay period?
They occur throughout the trial. We have definitely done versions without a delay period, although I don't think we've analyzed them as extensively as this. The reason we put the delay period in there, and I think it ends up serving a super important function, is that presumably the animal has made its choice definitely by the end of the stimulus period, probably even pretty early in the stimulus period. So by having that delay period, we separate the moment in time when the animal makes the choice with the moment in time that he makes the movement response to actually report the choice. So that's why we built in the delay. But you're certainly right that they're starting to anticipate their reward, and so there are a lot of temporal dynamics during that time that reflect fidgets and anticipatory movements of various sorts.
So just hold that thought for a second. I just want to tell people that we have a full house. There are people outside who are slightly annoyed that they can't get in. I want to make sure that everybody knows that there are various empty seats still. and including at least 5 here. I know this is going to sound mean, but we can't guarantee that after the break you're going to find your seat again, because there's a slightly larger number of people than seats, ever so slightly. Next person, overflow room. The overflow room is beautiful. It's got a big screen. Did you want to say something? Yes. Oh, you have a question. Just hold that thought, because we have a question there.
Hi, over here.
Yeah.
So this looks really great. I'm wondering to what extent you think some of the sources of these spontaneous movements are subcortical, and it's in response to the statistics of the environment overall. So if there is increased uncertainty, you might expect neuroepi to all of a sudden sort of increase gain in many places in the brain, and that could result into spontaneous movement, and it could also increase dopamine, and that could correspond to sort of increased vigor in movement. Neuroepi will sort of dilate the pupils. So a lot of things could happen if the animal thinks it's in a more volatile regime, or if it thinks this trial is too hard, is too much uncertain. Are you doing any kind of behavioral manipulation to test the effect of uncertainty and volatility of the environment and the sort of subcortical effects on these spontaneous movements?
Yeah, we're really open to the idea that these signals that are tied to motor regressors actually reflect internal state transitions, such as arousal or uncertainty. It'd be really nice to get to the bottom of that. For this particular study, the large data set that we have is with the one certainty level. I should say also, in terms of these being brain-wide signals, I think that's a really important point. So when we did the 2-photon, we only looked in area ALM, but there's another group, Carson Stringer and Marius Pachataru, who I mentioned a moment ago, also did very complementary work in V1, which I think really makes it clear that these kinds of modulation by motor variables, whatever they reflect, is quite widespread in the brain.
Okay, so we have 2 questions on this side. One over here. Yep.
Hello. First of all, thank you very much for the talk. It's beautiful, beautiful data.
Thank you.
I'm trying to reconcile what you just said and the data that was presented in the previous talk by Stan Dehan. So in the previous talk, the data suggested that you had these conscious decisions variable that's related to this huge ignition phenomenon that is visible all across the cortex, and in particular in the prefrontal areas. Whereas in your case, it seems that these decision variables, if existent, are extremely difficult to find, at least when you compare it to a random report. So I'm wondering whether you could speculate on what the differences are between the 2 paradigms.
I think there's 2 possibilities. So first of all, it may be that there must be decision signals somewhere in the brain. It can't be the case that our experts and our novices have identical neural responses, because if they did, their behavior would be the same, and their behavior is really different. So of course we know that there must be somewhere in the brain where their responses are different. And there's 2 possibilities. There's one possibility, which is that it's an area that we didn't look at, potentially subcortical, maybe the reticular nucleus.
Reticulus nucleus, I'm telling you.
It could be, I don't know. That's certainly one possibility. There's a lot of really interesting subcortical areas. I think there's a second possibility, which is that these signals are hard to see, and they are really only evident when you really clean up the data. I think if you really squint at our data, there are sort of a few signals in a few places that kind of hint that maybe that's the right place to look. And I really can't comment on other works, including my own. I think I myself measured decision signals in situations where I measured only really a small number of movement variables. But I think in a lot of this work, it really would be good to get some video hooked up so that other groups could start to understand how movements are impacting their signals as well. And it doesn't mean that decision signals aren't there, but they might be mixed with other kinds of signals, and they should be untangled.
Okay. Next question is still on this side by Jacob Yates.
Thank you.
Scream, then scream way louder. Way louder.
Okay, very cool stuff. I noticed there were lags in the design matrix, and I was wondering if you could say whether you think these movement regressors are predicting movements or reflecting movements that have happened, or if you have that resolution.
Yeah, so
maybe repeat the question.
Yeah, I will repeat the question. He said he thought the talk was awesome. And he had nothing to ask. No, that's not what he said. So he was wondering about the motor.
Oh, I see now.
About the time lag on the movement regressors. And indeed, like if there's a movement at time whatever, then we have a value in the design matrix exactly as you noted at both before and after that value, because there could be build-up activity. But actually, and Simon, who's here, can correct me if he disagrees. Really, it seems like most of the movement-related activity is around the time of the actual movement. So we did not see evidence for a lot of build-up signals, and what looked like build-up signals were often kind of related to ongoing movements rather than build-up activity for a particular movement. You agree? Okay, Simon gave me the thumbs up. So yeah, it seemed like we built that into the design matrix to allow for that possibility, but it was less prevalent than we had expected.
And real quick.
There's one more, yeah, back there, then maybe we'll stop there.
Thanks for a nice talk. And I'm very interested in why you choose a time or the perception of time as one of the features for a cognitive model. And I'm also interested in how do you manage to record, to accumulate this feature in respect to the mouse activity.
Yeah, so the regressor for time is really simple. It just has a 1 at every moment in time. And the reason we included it is because in other kinds of neural activity, it seems like there are both explicit representations of time, for example, in animals doing timing tasks, and it also seems that there are other signals that are intimately related to time. So one signal that I believe is in the brain, because it's important for interpreting behavioral data, is urgency signals. So that's a time-dependent rise that's independent of the stimulus of evidence, and that's the kind of signal that would be captured by that regressor. In fact, in this data set, as you saw, when we kicked out that regressor, it didn't change the model fit very much. So I think in these measurements, we don't see a signature of an urgency signal. And I think for people studying time, I think it's going to be important to consider these alternative explanations.
Oh, speaking of time, thank you so
Okay,
much
thanks,
for a
everyone.
wonderful talk.